Hello, and thank you for joining the Peach Education Conference as part of the 2021 Virtual Southeast Regional Fruit and Vegetable Conference. I am Andy Rollins, County Extension Agent with Clemson University, and will be your moderator for this presentation. Pesticide CEU credits and CCA credits are available for most presentations and are offered during the live Southeast Regional Conference. Check the Pesticide CEU guide CEU guide for a list of approved presentations and also participating states. The Pesticide CEU guide is located in the Event Resources tab under the Media Player. The guide is also available at the Resource Center located on the main menu of the conference platform. Please note that credits will only be available for registered attendees and only during the live conference. Credits will not be available for on-demand viewing. There is a simple three-step process in order to receive pesticide and CCA credits. First, you go to the audience chat box located on the left side of your screen and type your first name, your last name, and the states for which you're requesting credits. Again, go to the audience chat box located on the left side of your screen and type your first, last, and the states for which you want credits for. You need to do this for every presentation in order to receive credits. The second step is to sign out at the end of every presentation. And to sign out, you follow the same procedure. You go to the audience chat box, you type your first name, your last name, and the states for which you're wanting credits. You will be reminded to sign out at the end of the presentation. The third step is to complete the Pesticide CEU registration. You only have to complete this one time during the conference, and it is not required with every presentation. So to access this, go to the Pesticide CEU registration link, open the Pesticide CEU guide on the Event Resources tab located under the Media Player. The Pesticide CEU registration web link is located on the front cover of the guide. The presentation is pre-recorded to reduce technical difficulties. We will be answering your questions live at the end of the presentation. You can submit a question at any point during this presentation by typing your question into the question box and then pressing send. Don't forget to thank our 2021 sponsors and exhibitors by visiting the virtual trade show and the featured products pavilion. Lastly, don't forget to join us every morning for coffee chats and every evening for networking. Check the conference agenda for details. Next, I'd like to introduce Kendall Johnson with the University of Georgia, who will be presenting on phony peach disease in Southeastern research in commercial orchards. She is working primarily with phony peach disease and is a highly regarded expert in this field. Her master's degree is from North Carolina State University, evaluating glomerella leaf spot and fruit rot of apple. I look forward to hearing this presentation and what she has to say to us.
Thank you for that introduction. Again, my name is Kendall Johnson, and today my talk will be focusing on phony peach disease in Southeastern research and commercial orchards. So phony peach disease is a bacterial disease on peaches that was first observed in 1885 near Marshallville, Georgia by Samuel Rumpf. Samuel Rumpf is considered to be one of the founding fathers of the peach industry, not only in Georgia, but also throughout the Southeast. And that's largely due to the development of the Alberta peach. This peach was one of the most popular peaches for almost 100 years, as it was widely propagated and shipped to many different states. Now, this becomes one of the crucial parts of this story later on. So for almost 100 years, phony peach disease was thought to be caused by a virus, but then in 1980 was discovered to be caused by the gram-negative bacterium, Xylella fastidiosa subspecies multiplex. It's primarily vectored by the glassy-winged sharpshooter, and management options are often limited leaving growers to remove symptomatic trees as well as wild plum species as they can serve as inoculum reservoirs. Ppd symptoms include flat and dark and green leaves, shortened internodes, dieback, stunted tree size, as well as reduced fruit size and quality. In the picture on your right, you can see a Ppd tree that is short and stunted compared to the healthier tree on the left, or on your right. I wanted to talk a little bit more about this dieback symptom. As you can see, I've circled it in your left-hand image. And this dieback is often a less observed and less reported symptom as it is found later in the disease stage. So growers are often removing these trees before it can get to this symptom. We also began to see this bronzing coloration of the tree. And this was found later in September and early October, although this symptom has not been reported in the literature. And finally, we've been beginning to see this red spotting of the leaves. So this red spotting has also not been reported in literature. So we asked Dr. Libby Sinowitz from Clemson University to come test for viral diseases like PN, RSV, PDV, as well as for phytoplasmas, but all negative. So this could be a newly associated symptom with phony peach disease. So most Xylella-related diseases have this characteristic marginal scorching, like shown here, and bacterial leaf scorch of blueberry, Pierce's disease of grape, and pecan bacterial leaf scorch. But phony peach disease does not have this particular symptom, leaving it harder to detect in the field. So historically, visual detection is used to identify Ppd trees within an orchard, but visual identification is subjective and observer dependent. So I have 3 different objectives I'd like to talk to you about throughout this presentation. And my first is to compare Ppd molecular detection with visual symptom assessment in field. So in 2018, Drs. Chen, Bach, and Brannon developed novel primers and sampling techniques for PCR detection of Xylella in peach. They found that the root tissue was the only reliable tissue type for detection of the bacterium in the peach. So in January 2019, I was hired to further this research of this disease that hasn't been studied for almost 40 years. So from this paper, I chose 3 different primer sets for the to detect the presence of Xylella, as well as the 4th primer listed is to make sure that we are using peach DNA for every single sample. Also listed on this slide, you can see the Qpcr protocol I used, as well as the Qpcr detection limits. So a positive detection of Xylella would be having CQ values between 18 and 29. We were unsure if the CQ value fell between 29 and 31. And there was a negative detection of the bacterium when the CQ value was greater than 31. So to walk you through this process of how I ran Qpcr, I first had to dig up 263 root samples from a July Prince block at the USDA station in Byron, Georgia. Then I would strip the root to then cut small cross sections of the root and place those into a tube with 2 BBs. This would then, I would then macerate the tissue, and then it was prepped for DNA extraction using the Zymo Research DNA extraction kit to then use that DNA for Qpcr. So from my Qpcr results, I found that 38 trees in this block tested positive for phony peach disease. So this is the actual block that I tested with the rows listed at the bottom, and the number of trees tested was on the vertical axis. So the blocks in red are the phony peach trees, the blocks in white are healthy trees, and the black boxes are missing trees. So after we had the Qpcr results, I wanted to assess the visual raters of that same July Prince block, comparing it to our Qpcr results. So we used 10 different raters, 5 which were experienced, so they were familiar with these Ppd symptoms, and then 5 that were inexperienced, so they were instructed of those symptoms that morning. We had 2 different rating times on October 10th, 2019, in the morning and afternoon. And each rater used a scale of 0, meaning no disease, 1 meaning that symptomatic of PPD, and 0.5 if they were unsure. So looking at this map, I have the qPCR map on your left. So those were the lab results that I shared earlier. And then I created a heat map. So from this heat map, you can see 2 different colors, a red and a blue. The darker the blue means that the raters were more accurate when correctly assessing healthy trees, while the darker the red, meaning the raters were more accurately assessing trees as being symptomatic. So for example, I have boxed around a block of healthy trees shown in the qPCR results. And then you can see on the heat map that all 10 raters correctly assessed these trees as also being healthy. Then I have a block of symptomatic trees that were showing qPCR results as being positive, and only 8 or 9 of the raters correctly assessed these trees as being symptomatic of PPD. I also wanted to point out 3 different trees that no rater found or accurately assessed, which to me is showing that these are early detection as they're not showing symptoms. Peach trees often are not showing symptoms of PPD at least until 18 months post-infection. So after gathering all the rater data, we found that there was no significant difference between the morning and afternoon ratings, as the morning raters had 69.3% average accuracy and in the afternoon, 68.8% average accuracy. But we did find a significant difference between the experienced versus inexperienced. So the experienced had 75.7% accuracy compared to the inexperienced at 62.6% accurate. So if we wanted to look a little bit closer at the analysis of rater 1 in the morning, this rater had 71.8% average accuracy. So if we're looking at this in a disease management scenario, if we're roguing all the trees that were rated as disease that were actually healthy, we would be removing about 13.68% of this orchard, which would turn out to be around $3,700 per year. So if these trees had about 7 more years of potential production, this could be a loss of about $26,000. So then if we're looking at another scenario, so we're changing the uncertainty of those trees that the rater 1 were unsure of to being healthy, this rater's accuracy would increase by 12.6% to 84.4%. So what did we learn from this study? We found that the accuracy of visual assessment is highly dependent on the rater. Rater experience does improve accuracy. We could potentially be overcompensating, but we could also be leaving diseased trees to serve as inoculum. So moving on to my second objective, we wanted to determine the current prevalence of PPD and establish research in commercial peach orchards through on-site surveys in Georgia, Alabama, South Carolina, and Florida. From these surveys, I surveyed 17 different locations throughout these states where there are primarily commercial orchards, which are indicated in orange, but there were 3 research orchards at the USDA station in Byron, Georgia, a research orchard at Fort Valley State University, and then 1 at the University of Florida. At each of these survey locations, if I found symptomatic trees, I would take 10 healthy root samples and 10 symptomatic peach tree root samples to test for the presence of Xylella. I would also map a block of 20 by 20 trees and walk down each of these rows and individually assess each tree whether it was symptomatic or not. So to test my root samples, I used Agdia's Amplify-RP XRT Plus for Xylella, which is an isothermal DNA amplification and detection system. This system will give you rapid results between 1 and 2 hours, depending on how many samples you have. This was one of the best alternative detection methods I could have used, as I couldn't get into the lab this summer to test my root samples with PCR protocol. So to walk you through this process, I took small sections of root with a solution provided by Agdia and macerated the tissue to take the suspension and place into a small tube with a powder at 42 degrees Celsius for 20 minutes. After the 20 minutes, I'd place into this detection chamber that would give you one of two results. The first result is showing, or the first detection chamber is showing a positive result due to the control and test line both present, indicating the presence of Xylella, while the chamber on the right is only showing the control line, indicating a negative result, the absence of Xylella. So after testing all of my root samples, I found that I was 93.9% accurate when comparing the test kit to my visual assessment for healthy trees, and 95.5% accurate when comparing the test kit to my visual assessment for symptomatic trees. I was particularly interested in this orchard E, as I only correctly assessed 6 out of 10 of the trees as being healthy. So why did I fail in this situation? This could be because this orchard was a unique scenario. There was a high level of incidence at 30.5% for PPD. The grower is not removing these symptomatic trees, as well as peach often does not show symptoms until 18 months post-infection. So these are likely early detection of the disease. So now I have a Google Earth image side by side compared to my visual assessed map of that orchard E. So when looking at this map, a peach expert might think this field absolutely has armillaria due to the missing tree pattern. But Dr. Brandon and I searched all over this field and could not find a single sign of armillaria. So we're thinking these missing trees are due to PPD. I also wanted to point out that you can actually see these visibly smaller trees even from this bird's eye view. I've circled some of these smaller trees which match up to my visually assessed map as also having PPD. So from my survey locations, I found that all but 3 of the locations had phony peach, while the 3 that tested negative were in North Georgia and in South Carolina. So PPD was confirmed in Central and South Georgia, Alabama, and Florida, but was not confirmed in North Georgia or South Carolina, and this is likely due to the higher altitudes and colder temperatures. The PPD incidence in these sample orchards ranged from 12.12% to 30.5%, and the visual radar accuracy was 95.5% accurate based on the presumed true values of the AgDia test kit. So finally, my last objective is to determine the current prevalence of PPD in other states that were historically known to have this disease. So this is a map that was published in 1959 by Turner and Pollard, and it is showing the PPD spread. So the areas in the light gray are showing light but consistent spread of the disease, while the areas in the dark gray are showing serious economic spread. Not shown on this map were his reports in Pennsylvania and in Maryland. So using this map, we sent this phony peach state survey to 19 states that were historically documented to have this disease. We sent these surveys to peach and fruit key pathologists within each of these states. So some of the questions I would like to highlight from this survey is, have you ever seen PPD in your state? If observed, how prevalent was PPD when you observed it? And then on a scale of 1 to 10, how big of a problem do you perceive PPD to be in your state? So after receiving all of these surveys, the states that are with a red dot have not seen or have not had reports of PPD from 1970 to 2020. And the states that are that have the green dot have had reports between 1970 to today. Of the 6 states that are reporting the presence of PPD, Georgia and Alabama still remain prevalent to this day, while South Carolina only had prevalent PPD between 1970 and 2000. Texas was listed as not prevalent as they only had reports between 2000 and 2010, and New Mexico had a rare reporting in 2010. Florida, although listed currently as not prevalent, I can speculate that this may become a more prevalent issue with the increasing peach production within the state. So finally, I would like to wrap up this story about these grafting techniques. These grafting techniques were the same that were used in the 1800s as they are today, as well as the same layout as a peach nursery. So this is a picture that was taken at a peach nursery in Tennessee, but you can see how close these peaches are planted together. So because they're planted so closely together, this would be an optimal environment for a sharpshooter. They could easily move from tree to tree, as well as remain protected from predators. So if PPD was prevalent within this nursery, it could easily spread throughout. So in the 1900s, Hale and Rump were the two key fathers of the peach industry and had large nurseries throughout central Georgia. They would ship their propagated trees, like the Alberta peach that I mentioned earlier, to many different states. So they could have unknowingly be shipping these trees that are infected with PPD due to those latent symptoms I also referenced. So after 17 states reported infections, a federal campaign was put in place to stop the movement of this peach nursery stock from central Georgia. So from our historic knowledge and our current knowledge of this disease, I can speculate that PPD initially spread via central Georgia nurseries. And after the initial spread, distribution was likely reduced, not only due to the northern altitudes and colder temperatures that are not conducive for Xylella survival, but also due to the federal campaign and the destruction of symptomatic trees. Vegetation could also be a key component as to why the disease is still prevalent within Georgia and Alabama. So in conclusion, visual detection is highly radar dependent and therefore not always an effective management strategy. PPD is still prevalent in Georgia and Alabama, but not currently reported in other historically documented states. And on a scale of 1 to 10, survey respondents in Georgia and Alabama and Florida rated PPD as a 6 or 7, demonstrating it is prevalent. But in the remaining states, the respondents rated the disease as a 1 or 2, indicating that it is not an issue for their peach industry. So my future work is to develop a PPD identification training session for growers, disease scouts, and extension agents, as well as create a fact sheet. I would also like to identify disease distribution and spatial patterns within these surveyed fields, as well as utilize hyperspectral imaging for the early detection of PPD. So with that, I'd like to thank my advisors, those at the USDA, as well as a special thanks to those who helped me in my on-site surveys, as well as all the growers that led me into their orchards and my funding sources. And with that, I would like to take any questions.